Hello everyone, welcome back to our channel, and today we are going to discuss principles and methods of food preservation. So let's start with principles of food preservation. Food preservation aims to prevent spoilage, maintain nutritional value, and extend shelf life by controlling factors that lead to food deterioration such as microbial growth, enzymatic reactions, and physical or chemical changes. The main principles are inhibition of microbial growth, enzyme inactivation, moisture removal, oxygen exclusion, and chemical preservation. So let's see one by one what are these. Okay. First one, inhibition of microbial growth. By altering conditions such as temperature, moisture, and pH, microbial activity is minimized, reducing the risk of spoilage and foodborne illness. Second one is enzyme inactivation. So enzymes present in food can lead to the spoilage, color change, or texture degradation. So inactivation of enzymes, usually through heat or freezing, helps to retain the food quality. Third one is the moisture removal. Reducing water content inhibits microbial activity as microbes require moisture for growth. Next one is the oxygen exclusion. Limiting exposure to oxygen reduces oxidative reactions, preserving flavor, color, and nutrients. The last one is chemical preservation. Adding preservatives helps inhibit microbial growth and prevent spoilage. Let's discuss some of the methods for food preservation. Number one is heat preservation. The principle for heat preservation is heat preservation destroys or inactivates harmful microorganisms and enzymes that cause spoilage. So here we are using heat as a measure to remove or inactivate the microorganisms and enzymes present in the food. Okay, the process is usually widely used because it can be applied to various food products, effectively extending shelf life. Now, two types of heat preservation or heat processing are um, employed usually, and these are blanching and sterilization. The blanching is briefly immersing food in hot water or steam and used for vegetables to inactivate enzymes before freezing. Okay, and the temperature we have used here is 85 to 100 degrees Celsius for 1 to 5 minutes. Primarily they are used for vegetables to inactivate enzymes before freezing. Okay, next is sterilization. Sterilization is a high temperature process that kills for all forms of microorganisms. Microorganisms, creating a shelf-stable product and commonly used in canning. Okay, and here the temperature is 121 degrees Celsius for 15 to 30 minutes in pressure cooker or autoclave. The advantage of this process is it is very simple, effective in destroying pathogens, and the limitation is high heat may reduce nutritional contents and alter taste and texture of the food. The second method is pasteurization. Principle for pasteurization involves heating food at low temperatures then sterilization. So usually below boiling to kill harmful bacteria. Okay, usually this method kills the pathogens while preserving the sensory and nutritional quality of the food. Types of pasteurization involves LTLT, that is low temperature long time. Low temperature 63 degrees Celsius and long time 30 minutes. And this method is often used for milk. Second is HTST, that is high temperature short time. Okay, the temperature here is 72 degrees Celsius and time is 15 seconds. That is a short time period, right? So commonly used for milk and juice. Next method is UHT, that is ultra high temperature. Here the temperature is 135 to 150 degrees Celsius for 2 to 5 seconds. So here the temperature is very high and the time is very short, right? HT, the UHT or ultra high temperature, producing shelf stable products without refrigeration. And after this UHT process, usually the product is packed under sterilized conditions for the for preventing the exposure to microorganisms. Okay. Next, the applications involve used for milk. We know that pasteurization is usually used for milk. You also for juices and some canned foods. The advantages of the pasteurization includes maintain quality while making food safer. Okay, so the quality is maintained while the food is safe to be consumed. Different methods allow flexibility for product types. Now, limitation does not kill all microorganisms. It only kills the pathogens and requires refrigeration for some pasteurized foods. So if they ask which pasteurized food can be like can have a stability without refrigeration, it is ultra high temperature used in pasteurization method. Okay. Next method is canning. And the principle of canning is canning involves placing food in containers or sealing them and then heating them to destroy microorganisms. This creates a vacuum seal that prevents recontamination. So here the food is what they are sealed in a container and then they are heated to destroy all the microorganisms present in them. Okay, and this process creates a vacuum seal that results in the prevents the food from recontamination. Okay, next types of canning, mainly two types of said, that is one is water bath canning and second is pressure canning. So water bath canning used for high acid foods. Okay, so you need to remember that water bath canning is used for high acid foods and the pH here is the pH of the food or high acid food is below 4.6. Okay, and the food such as fruits, pickles, etc. are sterilized by this method, water bath canning. Okay, where the lower pH aids in microbial inhibition. Okay, now processing at 100 degrees Celsius for 20 to 45 minutes. Okay, next is pressure canning. Pressure canning is used for low acid foods, that is above 4.6 pH, okay, like vegetables and meat, and achieves higher temperatures like 115 to 120 degrees Celsius for 20 to 90 minutes to destroy spores of Clostridium botulinum, which causes botulism. Okay, so pressure canning is somewhat similar to the sterilization. Here the food is inside the cans. Okay, now process, the food is cleaned, sometimes blanched, packed in jars or cans, then sealed and then heated. Okay, advantage of this process, it produces long shelf life without refrigeration, ideal for emergency food supplies. Okay, and limitation is time intensive, high temperature can degrade nutrients and texture of the food. Next process is dehydration. The principle here is dehydration removes water. From the name itself, we can understand that it removes water from the food, inhibiting microbial growth and slowing enzyme activity as microbes require moisture to thrive. Okay, the method involves sun drying, hot air drying and spray drying. Okay, sun drying inexpensive but may lead to uneven drying and risk of contamination will also be there. Temperature, it depends on the climate, right? Next is hot air drying. Uses heated air to dry foods more consistently. Okay, he uses air. Heated air is used here. That is hot air. Said as we said, it uses heated air to dry food more consistently, and the temperature here we use is 50 to 90 degrees Celsius. This is suitable for most of the fruits and vegetables. Next is spray drying. Okay, often used for liquid foods. Okay, spray. Something is sprayed here. So for spraying, we need this food to be liquid, right? It is understood. So it is done in the case of milk. We have milk powders available in market, right? So they are spray dried. Okay, sprays the liquid onto hot air, causes it to dry instantly. Okay, and here the temperature varies from 150 to 200 degrees Celsius, and primarily for liquids like milk or coffee. Next is the application, commonly used for fruits, vegetables and meats. Dehydration is commonly used for fruits, vegetables and meats, as well as milk also. Okay. Next is advantage, lightweight and compact storage. When the water is removed from the food, it becomes lightweight. It will compact in structure so that storage will be easier. Ideal for dry shelf stable products. Okay, next is limitations, require proper storage to prevent moisture reabsorption and can alter texture, taste and nutritional quality of the food. Okay, next is freezing. So principle here is freezing reduces the temperature to a level that slows microbial growth and enzymatic activity. Although freezing does not kill all microorganisms, it significantly inhibits their growth. Now types, slow freezing and rapid freezing types are there. Slow freezing forms larger ice crystals which may damage cell structure and affect the texture, and usually the temperature is minus 18 degrees Celsius. Next is rapid freezing, forms smaller ice crystals, better preserving texture. Okay, so rapid freezing forms smaller ice crystals which preserves the texture of the food, and here the temperature is minus 30 to minus 50 degrees Celsius. Okay, now application used for meat, vegetables and prepared meals. Advantage, retains taste and nutritional quality well and widely applicable across food types. Limitation, requires continuous freezing. Thawed foods may lose quality or spoil quickly. Next is freeze drying or lyophilization. Freeze drying involves freezing food, then reducing the pressure to allow the frozen water to sublimate directly from ice to vapor, removing moisture without passing through the liquid phase. So what we are doing here is we are like reducing the pressure here to allow the frozen water to sublimate. Okay, we freeze the food first, then we reduce the pressure and we will what sublimate the food, that is directly the ice the food the water contained in this food is directly converted into vapors. It will not be like it will not be liquefied and then vaporized. It is directly vaporized. Okay, and this prevents the spoilage of the food. And the process here is food is frozen, placed under a vacuum, and heat is applied to allow ice to sublimate. Okay, now food is frozen at 50 degrees Celsius or lower. Okay, first they are frozen and then placed under vacuum to sublimate water, and the temperature during the drying may increase. So we have, as I said, it is the ice or the frozen thing is to be sublimated, right? So the temperature here is they increase the temperature to sublimate, and the temperature here will be 20 degrees Celsius or higher under reduced pressure. The advantage of this freeze drying technique or lyophilization is preserves texture, flavor and nutritional content exceptionally well, very lightweight. Okay, next is limitation, high cost. So this process is costly, requires specific equipment and proper packaging to prevent moisture reabsorption. Next is fermentation. Fermentation uses controlled microbial growth to convert sugars into organic acids, alcohol, gases, which acts as natural preservatives. Okay, so the types include lactic acid fermentation and alcohol fermentation. Produces lactic acid. Lactic acid fermentation produces lactic acid used in yogurt, sauerkraut, pickles, etc. And temperature range used here is 15 to 30 degrees Celsius. Depending on food type, like in the case of yogurt, it's fermented in the at 43 degrees Celsius. Okay. Next is alcohol fermentation. It produces ethanol used in wine, beer, and bread. For beverages like wine, typically the temperature used is around 20 to 25 degrees Celsius. Next is advantages. Enhances flavor, adds nutritional value, increases shelf life and requires minimal equipment. Limitations of fermentation says taste and odor may not appeal to everyone. Okay, must be carefully monitored to avoid contaminations. Next is microwave processing. The principle involves microwave processing uses microwave radiation to produce heat inside the food, rapidly destroying microorganisms through thermal energy. Application: pasteurization of food, soft food, ready-to-eat meals, and drying herbs or spices. Temperature generated varies widely, often reaching 100 to 200 degrees Celsius within seconds, and the exact temperature depends on the microwave power and food moisture content. Advantage of this method is rapid heating, minimal nutrient loss compared to other high heat methods, and the limitations involve uneven heating may lead to survival of microorganisms and not suitable for all types of food. Next is irradiation. The principle involves exposing food to ionizing radiation such as gamma rays or electron beams to destroy microorganisms, insects or parasites without significantly heating the food. Application extends shelf life of fruits, vegetables, spices and meat, and advantage does not affect temperature, allowing preservation of heat-sensitive food, improves the shelf stability without refrigeration. Next is limitation. Consumer acceptance can be low due to concerns about radiation, requires regulatory approvals. Next are chemical preservatives. Now, principal adding chemical preservatives inhibits microbial growth and enzymatic reactions, helping to preserve food without requiring drastic environmental changes. Common preservatives like benzoates, nitrates or nitrites and sorbates are used, and benzoate effective against yeast and mold in acidic food. Nitrates or nitrites used in processed meats to prevent botulism. Sorbates commonly in dairy or baked fruits products to prevent mold and yeast. Advantage: widely effective at low concentration, extends shelf life. Now, limitation: potential health concerns at high concentrations, strict regulations on usage levels. Next one is aseptic preservation, and the principle for aseptic preservation includes in here the food and packaging are sterilized separately and combined in sterile environment, ensuring a contaminant-free product. Process involves sterilization and filling and sealing. Okay, sterilization, the food product is sterilized often by UHT processing, and then sterile food is filled into sterile containers in a sterile environment. Okay. Now, application primarily used for liquid foods such as milk, juices and Soaps. Advantages: produces shelf stable products without the need for preservatives or refrigeration. Limitation: requires sophisticated equipment and precise control to maintain sterility. This is a quick summary table for revising the whole topic. So here we have divided the method with temperature, duration, applications and examples, and the notes. Okay. So blanching, the temperature here is 85 to 100 degrees Celsius. The duration is 1 to 5 minutes. Applications or example applicable for vegetable before freezing, and the main purpose of this blanching is enzyme inactivation. Now next is sterilization. The temperature here is 121 degrees Celsius. The duration is 15 to 30 minutes. This is mainly used in canned foods, and complete microbial destruction is possible by sterilization. We have to remember all these temperatures and the duration and which type of food is used for the competitive exams. So this table will be very useful. I hope you all write it down for your future reference. Okay. So next one is LT pasteurization. The temperature here is 63 degrees Celsius. Duration is 30 minutes. Milk and pathogen reduction needs refrigeration. HT pasteurization, 72 degrees Celsius. Duration is 15 seconds. Applicable for milk and juices. It retains the flavor and nutrients of the food. Okay, next is UHT pasteurization. Here the temperature is 135 to 150 degrees Celsius. Duration is 2 to 5 seconds. Shelf stable milk or juices, and no refrigeration needed if sealed properly. Next one is water bath canning. The temperature here is 100 degrees Celsius. The water bath or the boiling water, you can remember like that. Okay, the temperature here we use is 20 to 45 minutes. It is used mainly for high acid foods like fruits and pickles, you can remember. Okay, and next is used only for high acid foods. You have to remember, if somebody asks you or the question comes like which is the method used for high acid food preservation, um, exclusively used for high acid food preservation, we can say it is the water bath canning. Okay, now pressure canning is 115 to 120 degrees Celsius at 20 to 90 minutes, for applicable for low acid foods like vegetables and meats, and essential for botulism prevention. Next is sun drying. We know the temperature is ambient. Mostly it is like 30 degrees Celsius. Duration is days. Okay. Applicable for fruits and vegetables. Notes, if you say it is low cost, risk of contamination is there. Okay. Hot air drying, 50 to 90 degrees Celsius. It takes hours to dry things, and it is applicable for fruits and vegetables. Consistent drying and vegetable widely used. Okay, next is spray drying, 150 to 200 degrees Celsius. Within seconds it happens, and it is used for milk and coffee. Instant drying of liquids. Slow freezing, minus 18 degrees Celsius. It is variable, duration is variable. Home freezing, applicable for home freezing and common for most household freezers. Okay, next is rapid freezing, minus 30 to minus 50 degrees Celsius. It is also, the duration is variable, and commercially frozen foods are rapidly freezed and forms smaller ice crystals. Preservation preserves the texture of the food, while slow freezing forms larger ice crystals and preserves doesn't preserve the texture of the food. Okay, next is freeze drying. Temperature is initially it is minus 50 degrees Celsius. It takes hours, and this is the lyophilization technique. Okay, freeze drying, it can they can ask which is the freeze drying technique, or they ask that what is the other name of lyophilization. You have to remember that freeze drying is lyophilization. Okay, and here the duration is hours. It is applicable for food items like coffee, fruits, instant meals, etc. And vacuum assisted sublimation and preserves nutrients. Okay, sublimation method is used in which method? Such kind of questions can be there. Next is lactic lactic fermentation. That is lactic acid fermentation is usually done at 15 to 30 degrees Celsius, and the duration varies. Applicable for yogurts and sauerkraut, and no, the natural acid productions are seen. Now, next is alcohol fermentation, 20 to 25 degrees Celsius. The duration here also it is variable, and it is mainly used for wine and beer, alcohol. Okay. Then now it produces ethanol and carbon dioxide. Microwave processing, 100 to 200 degrees Celsius. It takes only seconds. Ready to eat meals or herbs. Rapid heating needs careful control. Irradiation, the temperature is not applicable here. Uses radiation, seconds to minutes. It is mainly applicable to spices, meats and fruits. Non-thermal method for shelf life extension. Next is aseptic processing, 135 to 150 degrees Celsius, 2 to 5 seconds. Milk or juice packed in aseptic containers. Okay, requires sterile packaging environment.